What I'd like to do is just give everyone, I guess, a little bit of a feel of what we're doing here in Australia with the Animal-Borne System Program or Animal-Borne Sensor Program, and then how that kind of fits in with the broader GOOS, so the Global Ocean Observing Network, ANIBOS, that we have, and how that might help us, or how that might feed into I guess, an integrated system of observing as part of the SOOS initiative, and where we can go with this in designing a system that allows us to observe in all areas, I guess with the emphasis on the fact that if we're going to be successful in observing the oceans, and the Southern Ocean in particular here, we need some sort of integrated complementary system, and hopefully after this, you'll have a nice idea of, I guess, the contribution that the animals can make to that vision. I'll skip through the slides relatively quickly because many of you have probably seen it before, but if there are specific questions, then please do sing out. And the other thing is that, like Danny said afterwards, we've got a couple of sort of questions, comments, and I think we really value your input into those 2 points, because I think that's really going to help drive where we go with the actual design of the system. So I'll get on to that. Like Danny said, my name's Clive McMahon. I work at the Sydney Institute of Marine Science, where I run the IMOS animal tagging program. Our main focus is to attach miniaturized CTDs to animals, primarily in the Southern Ocean. We do some tropical work in Northern Australia on turtles, but our main focus is attaching the instruments to seals in the Southern Ocean. Just briefly, the ANIBOS network was endorsed by GOOS, Global Ocean Observing System, in 2020, and as a UN decade project in 2021. Primarily, we collect subsurface temperature and conductivity measurements, of course, related to water column structure. I'm not going to read through our vision and our mission. This is all in our paper published a couple of years ago in Frontiers in Marine Science. If anybody wants a copy of that, please sing out and I'll happily give that to you. So a lot of the information I'm going to present here is actually in that paper. So don't get too caught up, I guess, in the detail. But, you know, overall, what we're trying to do is we're trying to collect good information from the Southern Ocean in particular and make that freely available to the community. So I will add here that that the IMOS vision is that all of the data we collect are made freely available to the community all of the time. So that's both in real time and in delayed mode. So the real-time data tends to go through the GTS and is available on World Ocean Database, and delayed mode data is made available on the AODN site, which is the Australian Ocean Data Network, and also on MEOP. Again, the sort of details of that are in our paper. Just briefly, what we're trying to do is we're trying to provide good information to the community that meets our societal and scientific needs to understand climate and the ocean. Of course, we want to set priorities for integrated observing systems, expanding that system to be more global and complementary. We like to think of what we're doing as cost effective. Yeah, the cost of the instruments on the animals, while still relatively expensive, are cheap in comparison to many of the other hardware that we would put in the ocean. And of course, we want to do this and make the data available to everybody, specifically coastal communities that are probably most vulnerable to changes in the ocean structure through sea level rise, etc. How do we do this? It's fairly straightforward. This is probably not hugely, or this is probably not great news to many of you. We have a miniaturized instrument. So we've taken a rosette that you might stick on, stick over the side of a ship. It's been miniaturized to a relatively small instrument that weighs about 580 grams, that we're able to attach to seals and turtles. There's a whole lot of sort of pre-calibration preparation of the instruments that happens in the lab. They then go off into the field, get attached to animals, and the animals then go off into the ocean, and each time they surface, they'll send through the Argos constellation information back through Toulouse, onto the Sea Mammal Research Unit in Scotland, where the information is decoded, and it's then made available to the community through the GTS, so the Global Telecommunication System. In real time, these data are used for operational forecast purposes, and in delayed mode for more kind of research applications. Oops. Okay. Just kind of where and what we're doing. I think the really important thing on this slide is really, I don't want to go into too much detail here, but you can see from the figure in the top left there that we do cover a large part of the ocean, and the little figure on the right really shows where we're concentrating our observations and where we're concentrating our effort, and that's mainly in the polar regions. And essentially what we have there is the blue line is the contribution that the profile contribution that the animals are making, and in the polar regions, by far the most profiles being returned are from animals and not from the other systems. Our main focus has been a time series, building a time series in the Southern Indian Ocean, and I'm very happy to report that the last of our CTD tags went out a couple of weeks ago from Eels Cabulin in the Southern Indian Ocean, and we're very proud of this effort, because this year represents the 20th deployment. So we have a 20-year time series this year, and we're very pleased with that, and we're hoping that that'll continue. So in the Southern Indian Ocean, we have now a 20-year time series of continuous sampling. Those red boxes in the top left-hand figure really just show Australia's contribution, but you can see that we're getting circumpolar coverage in the Antarctic or the Southern Ocean, and we're getting some really nice coverage in the Northern Hemisphere too, with little bits and pieces now starting to dribble in from the tropics. But I'm not going to go into that too much here. The bottom panel of the figure really just shows kind of, like the observing systems, one system isn't going to cover all of our needs. So different animals in different regions will provide the information that we need. So, we're not going to stick things to fish in the Antarctic. We're going to use seals there. We use turtles and sharks and other animals for the tropical observations. Our main sort of focus is collecting, like I mentioned, conductivity and temperature, but we are also able to sort of design and I guess tailor the instruments to collect other information: fluorometry, light, dissolved oxygen, wind, pressure, and various other bits and pieces that help us, I guess, observe the ocean in more detail. But the core program is the conductivity, temperature, depth profiles. This is just a little bit of a slide to kind of, I guess, give everyone a feel for what we're doing and where we're going with this. So I mentioned we have a core program, which is the CTD program, with some other bits and pieces coming on after. So fluorometry, light, oxygen. We have accelerometry and a magnetometer, so we can look at orientation and we can reconstruct movements of animals underwater. There's some passive acoustic measurements. There's high resolution information, so the instruments are bigger on those. We tend not to use too many of them, but they do have a sort of a niche that that we're looking at. And then more recently, we've got some active acoustics where we're attaching a sonar pinger and integrating that with the instrument to look at prey fields and integrate feeding behavior and prey fields with physical structure in the ocean. So I think that's a really nice example of actually quantifying performance of the animals directly relevant to the structure of the ocean, to provide a more synthetic view of I guess the environment and how that affects behavior and animal populations. This is just a little slide to show our core area of operation from the Australian sector. So here you can see the focus in the Southern Indian Ocean and a little bit of focus towards the Southern Pacific. We're hoping that as part of the new East Antarctic monitoring program that the Australian Antarctic Division is running, that we'll be able to sort of fill that gap in sort of the more eastern sector of the Antarctic a bit better, and start building a time series from Macquarie Island as well, which will give us, you know, really nice coverage towards the Ross Sea and then a little further to the west from there. So you know, this is just a conglomerate of all the tracks over the last 20 years really, and this represents probably in the order of 600 or 700 animals carrying instruments. I will say here that the Argos transmissions allow a return of about 4 CTD profiles per tag per day. So we do amass enormous amounts of information quite quickly each year as we deploy the instruments. And I'll talk a little bit about that in terms of how it fits in with the sort of broader observing systems or observing network when I talk about some of the effects of the pandemic on goose. I'm going to skip through these really quickly. This is just to show you that we do get some really nice data from those various integrated sensors on the tags. So here's some you know, just some images of the fluorometry. Of course, fluorometry on its own doesn't tell you everything. You kind of need to integrate light into that to get a better idea of the actual chlorophyll and the biological activity in the water column, and adding light, of course, gives you a much better idea about that. So you know, I guess don't get too caught up in the detail, but you know, we get some really nice measurements from that. This work is largely led by our French colleagues at CNRS in Chizé, Christophe Guinet and his group, who we've been working closely with over the last couple of decades. Dissolved oxygen, one of those things that that we'd all really like to measure in the ocean, but is actually not as easy as we'd like it to be. We've had 2 bites at the cherry on this. There were some early deployments of an optode. It worked relatively well. So that pink box is just the area up above there. So you do get some nice information. But there were some significant issues with drift and calibration of those sensors. So we sort of abandoned that program. And if you think back to that little timeline, you would have seen a gap in the dissolved oxygen measurements. So we've had another bash at it. Oops. There we go. And this is just some data from a new sensor that's been attached to a new dissolved oxygen sensor that's been attached. These are still raw data, so they do look a little bit messy, but this is kind of hot off the press, really. This is some work done by Christophe and his colleagues from Peninsula Valdez in Argentina. And it's looking really promising, the data that are being returned. So I think the key message here is watch this space, and hopefully there'll be some nice information coming out fairly soon, describing the new oxygen sensors and what we can do with those. So I think this holds some really nice promise for us. One of the things perhaps a little bit to the side here is that here in Australia, we've been leading a project to use the animal dive depths and animal behaviour. Many of the seals are benthic divers to look and map bathymetry. So some early work by Laurie Padman and Dan Costa off the West Antarctic Peninsula showed that diving behaviour and dive depths can be used really successfully to map bathymetry. Those are the top two panels of the figure there. More recently, this is a paper we have in review at the moment. We've taken all of the dives from the East Antarctic sector where we have information. The red bits in those two panels are where the seals are diving deeper than the interpolations or the bathymetric products. So there are lots of areas where that's happening. The bottom panel is an area around Casey Station, the Vanderford Glacier. In about 2010, we had an instrument attached to a seal, and the seal was consistently diving more than 1,000 meters deeper than, it's the wrong word, but the known bathymetry. What I'm getting at there is the prediction or the interpolation of bathymetry in that region. We were quite concerned about that. We thought that we were having problems with the actual pressure sensors. Turns out that the new Australian Antarctic vessel, the Nuina, managed to do a multi-beam survey there a couple of years ago, and lo and behold, discovered this very deep trench off the Vanderford Glacier, and this really led us to pursuing this idea of, can we use animals to map in more detail bathymetry, and it turns out we can do that. So hopefully I'll be able to share with the community our paper on this in the not too distant future. But I think this is a really exciting development for us, because bathymetry is such a key to understanding the processes, or sort of the oceanographic processes around the Antarctic coast, and also the interface between ice and water, so the ocean. So we're really excited about this component, and I think this is really going to start a whole new area of I guess observing for us. I will add here to the side that some folks in Scotland, off the west coast of Scotland, are using skates to map bathymetry, and a group in the US are using cormorants to do the same thing. So I think there's a lot of promise in the animal-borne sensor world to look at bathymetry. And given we know, less than a quarter of the world's oceans have been mapped properly, or in detail, perhaps not so much properly, but in detail. So, there's a lot of work for us to do in that space, and I think animals can help. Here, just a brief sort of description of the sonar technology and integrating animal behavior with that. We were able to detect quite nicely prey fields in the water column, and we can look at both seasonal and annual shifts in that productivity, and relate that to water column structure. Primarily the work we did in that bottom panel was looking at seal behavior. But again, probably not for us to dwell on too much here. Antarctica is fantastically interesting and important, but the tropics and subtropics are important too. And as part of the broader ANIBOS program, there's a big push to observe these tropical and subtropical oceans. We're using turtles primarily as the animals to capture our system here. And I just want to give you a little video here of a recent set of deployments in Northern Australia on flatback turtles, just to give you an idea of the kind of coverage we get from turtles. So they too move around and will return and collect and return data via the Argos system. So we get some really nice spread across Northern Australia, and of course, understanding what's happening in the tropics. They're poorly observed, but animals are helping us deal with that. So I guess this is really the nub of where we're going to go. We want to have an integrated system that serves all of our purposes and provides information across a broad spatial and temporal scale. Animals can do this with our miniaturized instrumentation, and they can help us sustain observing systems. And this is kind of, I guess, the point I was trying to make a little earlier on. Tim Boyer out of NOAA has led an initiative, and this work has just been published in the Bulletin of the American Meteorological Society. I guess really discussing the vulnerability of the global ocean observing system to things like the COVID-19 pandemic. You know, when people can't move around, we're unable to service many of the instruments that we need for returning observations. The systems that are reliant on people more directly, you know, deploying Argo from ships or servicing mooring lines, are more vulnerable than some of the other systems. And I think, you know, what came out of this work was that animal-borne system is fairly robust, because quite often animals are accessible from sort of locations where people can still be without the vulnerabilities of movement and sort of restricting interaction or movements of people around the globe. Of course, a major problem for all of these systems are those things of production of sensors and chips. But I think the main message, or the take-home message here, really is that that if we want a continuous sampling or observing system, we need to use all of the tools at our disposal, and that includes things like animals and all of the other systems. It's not a one-size-fits-all. I think, you know, perhaps here, you know, diversity and I guess a diverse system is going to be the one that's most resilient. And I guess, just to use a biological, you know, analogy, for systems or populations to be resilient, you rely on diversity across those populations. And I think if we apply that same thinking to ocean observing, we'll be doing well, because that's going to allow us to keep doing what we want to do and to do it well. And so, yes, here just towards an enhanced, and I guess what I really mean here is an integrated, and perhaps you can even change enhanced and integrated with a, you know, a synthetic observing system. We do need to use all of the systems. They all complement one another. Like I said, it's not a one-size-fits-all. I think the community is mature enough to recognize that if we want to do good work, we need to be creative, and we need to be inclusive in what we're doing. So thanks very much, everybody. I think I'll leave that there. I will just like to say that, of course, you know, this work is relies on the collaboration of many people and institutes. Some of them are recognized there. And I think perhaps now we can open up the discussion with those two key points we have. Hopefully everybody received the email with those 2 points. So thanks, everyone, and I'll pass over to Danny.
Great.